And it's my very, very special pleasure to introduce our today's speaker, the speaker of the anniversary event here, Alfonso Martinez Arias. Alfonso is an old friend and colleague, a long-term friend and colleague, sorry. A well-established friend and colleague. No, and he's been here 10 years ago. And he has opened the entire series, so I'm super happy that he could be convinced to come here again and tell us his story 10 years later. What you need to know about Alfonso is, I think, he was a superstar in genetics in flies. But he had a question that went beyond that and then completely abandoned, I don't know whether completely, but in terms of publications, almost completely abandoned flies and moved to an entirely new thing. And he simply was interested and thrilled by the question, what does it take for a single cell? I don't want to give you a talk. What does it take for a single cell to develop rather than divide? And that's something that wasn't easily addressable in flies, but was addressable in vertebrate systems. And I'm pretty sure we will hear more about the power of cells. And I think for me, he's really a role model. You should follow your ideas and should not just stick to something where you've been successful at. And I think that is something for your life. You think you need to know what you have to do in your next step? Next 10 years, 20, 30, 50? No way. You will find out every day anew. And that's the way you will hopefully enjoy your life and you will be successful and make important discoveries, at least for yourself, maybe for the world, as we will see here with Alfonso. Alfonso, thanks a lot for being here again, and we're looking forward to your presentation.
Danke. So, mit meiner sehr schlecht Deutsch, ich möchte sagte Dinge. Ich bin groß sehr dankbar für die Gelegenheit, den Bertalanffy-Vortrag zum 10-jährigen Jubiläum zu halten und insbesondere jetzt und später heute zu Ihnen zu sprechen, um mit Ihnen über Wissenschaft, das Leben, was auch immer Sie sprechen möchten, zu sprechen. Es gibt keine größere Freude an meiner Arbeit, als die Gelegenheit, mit jungen Frauen und Männern zu sprechen, die an der Schwelle zum Start in ein unabhängiges Leben stehen, voller Neugier, Fragen, Hoffnungen und sicher auch Angsten. Um meine Erfahrungen zu teilen und ihre zu hören. Der Grund dafür ist, dass sie sehr wichtig sind. Die alle wichtigen Personen in dieser herausfordernden Welt, in der wir leben, repräsentieren sie die Hoffnung und die Wissenschaft, die sie heute hier erbringt, ein wichtiges Instrument, um sicherzustellen, dass die Hoffnung zur Realität einer besseren Welt wird. Ich bin sicher, dass einige von Ihnen dazu beitragen werden, die Wissenschaft als Werkzeug zu nutzen, um eine bessere Welt zu schaffen als die meine Generation Ihnen hinterlassen hat. Aber Wissenschaft ist nicht nur ein Weg, die Welt zu verändern. Es ist auch eine angenehme Aktivität, weil es darum geht, zu lernen, wie die Welt funktioniert. Und in diesem Prozess entdecken wir, dass Magie wahr ist, weil die Art und Weise, wie die Planeten und die Sonne kreisen, die Mechanismen, die Wasser erhitzen oder gefrieren lassen, die Art und Weise, wie wir heute sehen werden, wie Organismen gemacht werden. Magie in sich eingebaut haben und Magie, sie werden mehr zu stimmen, ist eine wunderbare Sache, die man beobachten kann. Die Wissenschaft enthüllt die Zaubertricks, die die Natur anwendet, um die Welt uns am Laufen zu halten. Heute möchte ich etwas von dieser Magie mit Ihnen teilen, wie das Stellen einfacher Fragen wunderbare Details darüber enthüllt, wie ein kleiner Teil der Natur der in Form und Form von Tieren verboten ist. Und jetzt bitte ich Entschuldigung, ich die Sprachen wechselt und Englisch sprechen, aber ich hoffe, Sie werden, wenn nicht alle, doch einige der Dinge genießen, die ich mit Ihnen teilen werde. So, Entschuldigung. So, I'll try to speak slowly, which, as Jochen knows, is very difficult for me. So, I don't know whether you've ever wondered how we are made. How many of you have ever wondered where do we come from, how we are made? You don't have to raise a hand if you've never asked that question. And how many of you think, just out of curiosity, because we live in a certain time, that we are made by our genes, that everything that we are is in our genes? How many of you believe that? Fascinating. So what else is there to do? What else is there to do other than genes? So this question has been asked by many people over the years, and you can see that here, where there have been many answers over the years about this. Some people think religiously this is a beautiful sculpture by Auguste Rodin, in which is representing the creation. Out of the hand of God, there goes Adam and Eve. Leonardo da Vinci tried to find this out. I don't know why this is not working, but the point, oh no, sorry about this, but Leonardo da Vinci tried to represent, tried to figure out what was inside the womb of people. Today is very normal for all of you to know that we come from inside the womb of our mother in some mysterious way, and that mysterious way was represented as in these in those things on the left. Ideas, more clear ideas about how we were made, how we were functioned, appeared in the 17th century because of this treaty that was published in France in the 1800s called the man-machine. The man-machine was an idea because at that time the physicists had discovered the laws of nature, people were starting to construct devices based on mechanics, so they started to see that perhaps human beings, organisms, acted like machines. And this is why inside there were a lot of machines. And they led people to start, some clever people, to start making up copies of ourselves as if we are machines. And one of the most remarkable ones, if you ever go to Switzerland, is this trio of people by a very famous Swiss watchmaker, Corpier Jacques Droz, who built these three, which are still in Neuchâtel, exactly as you see them there. There is a scribe, there is a draftsman, and there is an harpsichordist. The draftsman, it's an amazing piece of work because, as you can see here, it moves the eyes, it writes, it can write up to 10 different texts, it will erase the text when it's wrong, and it's probably the first programmable you can see there in the back of thing. I mean, Swiss are very good at making these precision instruments, and this was a very, very remarkable object. And that's the way people made this similarity. They thought that we were made of some natural copy of metal and wires and plates. This really got to a very interesting, this notion of the man as a machine got to a fantastic view by this man, who many of you don't know, called Fritz Kahn. He worked in Berlin, and over a number of years he published this work, Der Mensch als Industrialpalast. I mean, it's a very fascinating set of drawings which he put out in the form of booklets over a number of years, and represented the human being, as he says, as an industrial palace. I learned that somebody had made a movie putting into motion what Fritz Kahn did, and you can see it here, and I hope that it will work. You can see. We are breathing, that's the lungs. Now we focus on the heart, the pump that is pumping the blood. I think this is my favorite bit. What happens if you are an industrial palace when you eat? There is a bar of chocolate. There it goes, down your throat. I like this bit. It goes into the stomach, and you can see they're throwing all the acids, everything to dissolve the food. There is a man with a lever waiting for everything to accumulate. And then when everything is ready, he will pull the lever, and there it goes into the intestine. And there, the goodies are separated from the crap. You see, there the goodies are floating about, and they're going to be sent somewhere else, and the rest is going to the waste. And then we can get back, that's where the liver, the pancreas is taking all the goodies and translating them into something interesting. Remarkable, it's fun. There is a wee-wee going out after you've separated the things. And you know, the goodies go one way. Remarkable. So, in many ways, this idea of the man as a machine is the progenitor, the precursor of all these prosthetics that we know about. And really, a lot of things, some of those are wishful thinking, but some are real. And you know that many valves of the heart and many other pieces of our body are made mechanically out of this notion that human beings, there is something mechanic about the way we work. However, if you look at your skin, I mean, today we know that this is not the way our components is not wire and metal. Our components are other elements that we can watch if we get deep into our body. In the 60s, there was this movie called The Fantastic Voyage, which they made, it's an American movie in which they take people and make them really, really small, and they make this little vehicle, and then they inject them into the blood of the organism. And they start navigating in this machine, and what they see, it's cells. I mean, there goes the people. It's a very classic movie that is being made, and people navigate through the blood system, and they get into dangers in the different organs. And so what they see is cells, because indeed, if we look beyond the surface, if we get a bit deeper, that's there you have a situation of the, there is a delay, that's my problem with the, I can see now. So there you have the layers of the skin, and if we go even deeper and nicer, we can see this majesty of the cells. Because that's what we are made of. And as I will show you in a minute, it is the cells that make us in a very interesting manner. You can see here one of Jochen's favorite organs, which is the variety of eyes. I mean, the creativity of the cells is enormous, because if we look beyond that initial surface, we will see cells everywhere. And look at the variety of cells. And I'd like you to think, I mean, if you hear about it, you are hearing me because, again, these cells are arranged in these fantastic and these beautiful palisades, which are essential for your hearing and to distinguish all the sounds. So wherever you look, it's cells, because cells have a variety of shape, a variety of form, a variety of creativity, that if you think about DNA, and you've seen some of the pictures this morning that some of you have put, DNA is very boring. It's just the same thing. It doesn't have this kind of creativity or ability. And it surprises me, and this is a theme that I'd like to share with you, why we have fallen into the trap that we are represented in our genes in some form, when actually we are represented in our cells. In your body, you have 440,000 trillion cells. 40,000 trillion cells, that's what your body is. And what is remarkable is that those cells are changing all the time. So, for example, those numbers that I'm giving you there are absolutely true. You are making 2 million red blood cells per second. Each of you is making, your bone marrow is making 2 million. Think about it, every second. I mean, every time I see this number, I shiver. It's absolutely remarkable. And what is interesting is that, and Jochen knows that, an eye is forever, but not the rest of your body. In fact, everything in your body, except your heart maybe, and certainly your eyes, is turning over all the time. Sometimes, your skin, you get a new skin every month, so to speak. You get a new intestine every 2 or 3. Every part of your body is turning over. In fact, you are not going to be the same in 10 years' time. One of the things that excites me is that your skeleton will change every 10 years. It will be completely renewed. Why? That's because, when my clothes don't fit me, it's because my skeleton changes. Nothing else changes in my body. So I think that you have to appreciate that you are changing. You are a work in progress of your cells. And that throughout your body, who you are in terms physically changes. And I find that very remarkable. A lot of that work is due to the fact that in every organ of your body, there is a very special population of cells which are called stem cells, whose job is to do this renewal. I mean, we think because we see ourselves, we tend to think that we are something static, that we are something, but actually there is a lot going on. We're just fleeting structures, fleeting creations of ourselves. This is why I find very remarkable that today everybody is saying that you're going to find who you are, when you're going to live, when you're going to die, all in the DNA. I could spend, and maybe we can talk later, many examples in which that is not the case. You cannot pin down much of what we are into. And the problem is, and that's for you to solve in the next few years, that is the cell. Because it is all that is in the DNA, all that is in the DNA is instruction to make nanomachines. Proteins and the protein complexes are nanomachines. And those are in an aggregate in this sort of milieu of lipids and sugars, and they create cells. And in the cells, everything, once you get a cell, the cell controls everything. So it's not that the genes make the cells. Once you create a cell controls everything. Think of your nervous system. It's not your genes that is letting you listen, that is letting you think. It is the interactions between those cells. The genes are just providing the tools. And indeed, inside the cells, there is a lot of nanomachines, and you can see that in the next movie. Inside I mean, there are machines. This is a representation. But that's, as Jochen knows, a few years ago I was very interested, and I'm still in noise. How can we listen to the noise of the cells? There you have it. Very remarkable. And really, all these we have gleaned over the last 50 years, and everybody and many colleagues of Jochen and me have contributed. We think this is not a bad representation of what is going on in the cell. So not only your cells are changing all the time, but your cells are a constant sea of activity. And you know, we are going now out of the nucleus. You will see some of our favorite molecules, and some of you have seen them today. There is the nucleus. You can see mitochondria floating there. You see the Golgi apparatus. And many of you this morning in the cytoskeleton is a remarkable thing which shows this fleeting nature of everything in us. Because here you have, there are cables, there are scaffolding. This is what is the tubulin. We saw. And you know, there are those things. They actually move like that. They actually move like that. Okay, this has been shown and this has been seen. They have legs. Some of them, they carry cargoes. Remarkable. And all this has been, today you can see these things at the single molecule level. In fact, that has been the great revolution in biology, the ability to see these things. And you know, these microtubules that some of you, they are constantly making and unmaking. They are constantly in. So all this stability that we see, all this being there, sitting that I see, there is a lot going on, not only at the level of your cells turning over, but at the level of the molecules. I don't know whether they made that noise. This is the actin that some, you know, when cells explore those things, they're doing that. There's machines, we know that there is. So that's what the genes do. The genes code for the parts of these nanomachines. And when these machines assemble, they start doing things that genes cannot dream of. But that's a muscle contracting when you're running. So this is, I want you to get this idea that cells are hubs of activity. And that not only cells are hubs of activity, but we are also hubs of activity of ourselves, this idea. Now, this structure, this remarkable structure that is the cell, how did it come about? There are two kinds of cells. Oh no, now it starts. Well, that's not what I wanted. Should have done it before. The magic that I made reference to, magic happens all the time. So there are two kinds of cells. There are two kinds of cells that you probably have learned in school. There are the prokaryotes, and there are eukaryotes. The prokaryotes are the very simple. They don't have a nucleus. They have some DNA wrapped in the middle. They are simple cells, but don't be fooled by simpleness, because they've been here way before we arrived, and they will be here long after we are gone. So that's very important and very humbling to have. And then there is the eukaryotic cells, which are very complicated. They have so bacteria that don't have all those complexities that I've shown you there. So how this very complex structure, how did it come about? How did it come about? There was a woman called Lynn Margulis, who many years ago, when she was a young woman, she had a great idea. She noticed something very strange. You all know about mitochondria. And she had the thought that mitochondria look very, very much like bacteria. So he had the idea that perhaps what happened in the course of evolution is that the way a eukaryotic cell arose is at a certain point that there were many different kinds of bacteria, unicellular organisms. They were eating each other all the time. And in one of those eating situations, some bacteria that performed the same function as the mitochondria were eaten by another type of cell, and they started to cooperate. They figured out things to cooperate. And that, she thinks, is the way that in this merger, this is the way the eukaryotic cell arose. In fact, there must have been many of these mergers, because when the bacteria that were eaten by one of the cells were able to fix oxygen and were able to, that gave rise, there is all these bacteria which are called cyanobacteria. A cell that ate one of those became the plants. And cells that ate other bacteria that were better doing, not able to fix oxygen, that they were fixing other things, they gave rise to the animals. So these single cell complicated organisms, they sort of come, you can see here the branch that goes to the plants, and these other plants goes to animal-like structures, which are called protista. These protista are very, very remarkable. This is a drawing from a very famous German scientist, very great artist also, called Ernst Haeckel, who was very interested by this world. And look at this remarkable. A lot of those are single-cell organisms. Those are single-cell organisms. Look at the variety that there are. And some of them are absolutely very, very beautiful. This is some staining with some antibodies to reveal the internal structure. That's called a stentor. It's a very interesting and intelligent animals. I have a good friend in Harvard who is interested in the behavior of these simple cells. In fact, it has a very good story. This has fascinated the scientists for a long time. So this thing called a stentor sits on the floor, and it's waiting there. And if you go and put a little bit of ink on it, it will move like this to try to get away all those drops. If you start pouring more, it gets very agitated, and it starts agitating even more. And if you put even more, what it will do, it gets so agitated that it lifts itself off the ground and moves somewhere else away from the ink. So that says that it's thinking. Even a single cell can do that, okay? So that's very remarkable. And it is here, somewhere here, that the ancestors of the animals lie, in this world of the protista, of the single cell prokaryotes. Something very mysterious, and I was talking to Stephen this morning about that, something very mysterious must have happened here, and this is one of the great mysteries of life. How do you go from this? There is actually a whole group of animals, like here, of organisms, sorry, I should say organisms, which are not animals, but they behave in a funny way. They are much more complex than this, but they are not animals yet. An animal is a multicellular organism, and something must have happened at some point. You can see here some of these very interesting organisms that are not animals. They try to form colonies, they try to interact with each other, but they don't form these structures, these compact structures that are animals. At some point, about 600 million years ago, I know it's an amazing number, I'm throwing numbers at you, 40,000 trillion cells, 500 million years. I mean, when a physicist tells you about stars and how many stars are in the universe, you can laugh, because there is more cells in the world than there are stars in the universe at this very moment. So that's something that should humble them. The physicists are very afraid of biology, by the way. You should take very good. We are very impressed by the physicists, but they are very afraid of biology. This is something for you to think about. Ask them to explain life to you, okay? In case there is any physicists in the audience. But you can see these very interesting animals, which, organisms, I keep on calling, but these organisms are at the interface between the protista, these single-cell organisms, and the animals. And the first animals that appear, there seems to be a gap here. All of a sudden, you start seeing, when you look at fossils, you start seeing these organisms. You just start seeing sponges. Sponges are the first animals, and we know that they are animals. You also get these beautiful cnidarians. This is Hydra, that some of you know, and of course there is a lot of work going on here in Heidelberg on Hydra, and this is another cnidarian. This is called a ctenophore. They are comb jellies. They're absolutely beautiful. These are the first experiments. I mean, what we are looking is the remnants of experiments that nature has done. These are not quite like us, but They obviously occupied the Earth for a very long time, when mostly was submerged in water. And then, 500 million years ago, something remarkable happened. It's called the Cambrian explosion. Because all of a sudden, this was discovered in a little shale in Canada called the Burgess Shale. And all of a sudden, out of this, what you see before is these boring organisms, not a great variety of them. And all of a sudden, about 500 million years ago, you get a huge explosion, a huge experiment in animal form. You get a lot of weird things. I mean, this famous biologist, Stephen Jay Gould, has written a book called This Wonderful Life about this explosion. They found all these fossils with animals that some of them resemble some of what we are today. I mean, they have what we call bilateral symmetry. They had a head and a tail. There are others that are very, very weird. But it's clearly nature experimenting. Nature is experimenting here with form. And of course, it's not DNA that is experimenting. It is the cells that are experimenting with configurations, with activities, with things that will fool them. And out of this Cambrian explosion, we get something that starts looking, we get evolution going big time. And you can see here, I mean, you know, initially, that's a tree of what happens. I mean, what we are today, it's a consequence of this. All of a sudden, it accelerates. Once you invent cells, once you get the eukaryotic cells, they start exploring a very wide space. They start being creative. This is what I find. And indeed, you know, cells are incredibly creative. I mean, it is all down to the cells, and they create structures like this here. I mean, some of you talked about plants this morning. I mean, this is the eye of a fly. You can see here on the left. This is one of my favorite structures that you can see here. So what you can see there is a paramecium. This is a unicellular organism. And that's 200 microns, 200 microns, a fifth of a millimeter. That is a fly. That is not a fake. There is a fly, which is about 200 microns. And it has a nervous system, and it has eyes, and it has wings. And the length of that is 200 microns. You will not be surprised that its nervous system has thrown out all the nuclei. That little fly is called Tinkerbell Anana, from Tinkerbell in Peter Pan. So because it's really like Tinkerbell in many, many different ways. But you also get the beauty of the, I mean, it's just the creativity of cells. It excels anything that an artist can do. And those patterns and those shapes and everything. So how does all this come about? Well, because we are one cell becomes two become four, and so on, and then we become many. What you see in this movie here is actually a human fertilized egg. Now in vitro fertilization clinics, people are given movies of the eggs that have been successfully fertilized, and they like to think that this is the child. We can discuss that later, but they give them the movies. Yeah, I can see that. So there you go, you know, a cell becomes two to become, and in the process of 7 days, what you're seeing there is the first week of the life of someone. That's what it is, forms the blastocyst. You can see the movie is repeating, but the first thing that the cell does is to become many. That's the important thing first. And then, when there are millions, and in the process of being millions, the cells start becoming different from each other and doing things. This, by the way, is from a very wonderful book that we've had at home for many years called Cells Are Us, which is the title of the talk. Really, because in a way, to me, as you can see here, the development of an organism, that's what we are talking about. Jochen said, I mean, a fly, of course, comes also from a single cell, but it's a dictatorship. The fly is a dictatorship. This is a democracy. The way a vertebrate develops is a democracy. And you know, it's about cells. It's not about mapping genes into cells. It's about cells mapping into space and time. And genes are just instructions to make the component of the cells. And you can see the beauty of development in these movies. I just took a couple of random ones. Here you have a Drosophila embryo. The ones at the top are zebrafish. Jochen never gives me a movie of Medaka, so one day maybe he will, and I can put it in the gallery. And there you have a chicken making the soma. It's absolutely amazing. The first thing that the embryo does, or this cell, when it amplifies itself, now you get a whole ball of cells that are all the same, and then it's going to go through this process called gastrulation, which a very famous biologist that unfortunately passed away 2 years ago, Louis Wolpert, defined in that manner. The most important event in your life is not birth or marriage, or death, but gastrulation. You didn't know that, but you know that now. And why is that the case? And why is that the case? Because that's the moment that that ball of cells, that is the outcome of the fertilization, engages into a choreography, a self-origami game, which transformed that into an organism. This is a very famous Xenopus movie. I know there is a friend of mine in the audience that will be very happy to see this movie. And there you have, you have a lot of cells that are all the same, and all of a sudden this choreography starts, and out of that choreography, you get something that looks like an embryo. You can see why I say there is self-origami, or it is a choreography. In fact, the cells are moving in very defined ways. This is a drawings of a scientist on that. Something that you can see there, some cells are going inside, and you can see how from posterior to anterior you get the nervous system, which is outside. Inside there are the muscles. Now I'll tell you how you're never going to forget what gastrulation is, which is a sentence not mine, but of that famous scientist called Ray Keller. Gastrulation is the act whereby you separate the asshole from the brain. That's what it is. And in fact, that little hole through which, now you're never going to forget. Gastrulation is the most important moment in your life, and is the one whereby you separate the asshole from the brain. And as we all know, many people don't achieve that separation very well. So that's very good. So, sorry, I don't know why. Do you know any? Anyway. Not sorry. But the question here is, how do we, so we've gone these bionics, it's very good, but if the cells are all this creativity, can we actually change this bionic into something that we can make with cells? Can we start working, instead of with wire and metal, can we actually use it to build, can we use cells? How can we do that? I told you before that there are these stem cells that are in every organ of ours, but there is a very special kind of stem cell that doesn't really exist. We've managed to get it in a plate out of very early embryos, and it's called the embryonic stem cell, okay? This is the embryonic stem cell. Oh, sorry. And the embryonic stem cell, I hope it can stay there. It passes by. I'm going to try to freeze it here. I think, I'm sorry about that. Well, these embryonic stem cells, we get them out of embryos. We can culture them in a dish for a very, very, very long time. And they keep this ability to generate the organism while they are on the plate. And it's very remarkable. How do we know that? Because if we put it back in an embryo, they will generate a mouse. Okay? And this is very important. Now, many years ago, a scientist in Cambridge, in Oxford at the time, called John Gurdon, did an experiment that led to these other experiments many years later, which is that you can take any cell of your body and actually get it back to the first cell that was in the embryo. You can turn back the clock of the cells. And this was exploited by these persons, Shinya Yamanaka. John Gurdon worked with frogs. Shinya Yamanaka worked with human cells and with... And what he did is this kind of reprogramming, in which, as you can see here, you take cells from skin cells, and using a cocktail of transcription factors, of molecules, you can turn them back into embryonic stem cells, which then you can differentiate. That's what stem cells do, into any cell tissue of the organ. And now the idea is, people are working on this very actively now, you should be able to take these cells and use them as the source of those tissues and repair. And that's really what's being done these days in terms of the engineering. I mean, a lot of work is being done these days precisely about turning these cells into the different parts of the organs of the organism. We already know, for example, these cells, if you don't do anything to them, I don't know, it's taken, there is very little we can do, I know. But if you turn them, you can turn them into brains, for example. You can turn them into livers. You can turn them into eyes. It's a very, and we know, we are not sure. It's a bit of an alchemy. It's something that happens. And indeed, this reminds me that the lecture that I gave 10 years ago sort of had very big references to Harry Potter. And I looked at it and I say that I was talking about the alchemy that we can do to get cells to do that. So using embryonic stem cells, in the case of the mouse, we have been managed, together with other people, to reproduce the very earliest stages of development. And I put Harry Potter there because there is a lot of magic. It is for you guys who are interested in this to explain to us what is that is going on after all. Let's not look down into alchemy, because alchemy is the progenitor, is what gives rise to chemistry. So the alchemy is not bad, it's just very early chemistry. So using these kind of tricks with those stem cells, we've managed to reproduce the very early stages of development, but we were wondering about whether we could reproduce gastrulation, which is that moment in which we're going to change the whole mass of cells into something that shapes us. And we did that in the lab, and you don't need to worry about the details. We call these things gastroids, and you can see we put a very small number of cells, and the cells seem to know how many they are. And if we put the right number of cells and they all get together in the right time, you start getting, for example, you will see now a gene there that is starting to be expressed and it becomes restricted. We start with about 200-300 cells. And as you can see here, those 300 cells become these very, very remarkable objects that elongate. And when we look at the genes that they are expressing there, we see that they seem to have the same organization in gene expression as an embryo. In fact, as you can see here, these things acquire a coordinate system in which you have the three coordinates. You have the anteroposterior, the dorsoventral, and the left-right. This is in vitro. This is in a dish. So these cells have an ability to make something that very much resembles the organization of an embryo. You don't need to worry about these pictures. It's just to show you that the organization of the cells in these structures is very much that of the embryo, and that they follow paths of differentiation. There is a very special class of genes called the Hox genes that we all have, which seem to act as a ruler about what the different parts of the body have to do from anterior to posterior. So they are very special genes because they are very conserved. They are always expressed in like a Russian doll kind of pattern. And indeed, in these structures that we call gastroids, they have the same expression pattern. You can see that they have this nested expression. So these structures are very intriguing. And we can follow in them, that's the cells, we can follow all the different parts of the earth. We can make nervous systems, we can make muscles, we can make guts, we can make blood, we can make a heart. You don't need... So, but this is all integrated. This is really like an embryo. But some of you might have realized that this does not look like an embryo. Although it has all the genes perfectly organized, it's lacking something. And that something probably is some interactions between cells that are going to shape that into something that looks more like an embryo. Because an embryo, an organism, is not just a collection of genes. It's also a collection of cell interactions. And in this lab culture system, you are missing that component of input. This is a very famous set of drawings from Picasso. What we are picking up is the essence of the organism. It doesn't look like a bull, but it has many of the elements of a bull, and you can recognize that as the structure of a bull. All this work was done with mice. So we wonder what would happen if we could do it with humans and try to see what Leonardo did. And indeed, I mean, we managed to create these structures, which we call gastroids. This is from human cells. And they look very much like the same of the mouse. They have this very beautiful organization of gene expression. They are very reproducible. They happen very, very efficiently. And all you need to know, we get them to do all sorts of things, and we can do all sorts of analysis that you don't need to worry about. And when we look, because there are ways that we can say how old, what stage of embryogenesis, of the development of a human being, these structures resemble. And we realize that we have managed to reach about 21 days, which is at the end of gastrulation. So we are able to reproduce, well, not we, it's the cells. We've managed to communicate with the cells in a manner that we've encouraged them to reproduce the very early stages of human development. This is very important. Why it's very important? Because there is no way of studying human development. We can study animals, we can study mice, we can study fish, but we cannot study humans because we cannot do experiments with humans. So the fact that with cells we've managed to create a system that reproduces many of the early stages of human development is going to allow us to create and learn something about how the basic body plan, the basic structure that we are coming to being. And for example, we are reproducing this system, very special processes. Here you have a very famous process which is called somatogenesis. It's basically we are segmented and we grow from anterior to posterior. And during this time in the embryo, you lay down these masses of cells which are called somites, which will give rise to your ribs, to the muscles of your thorax, and we managed to reproduce this in vitro. You can see there how this is one of these gastrulates, and you can see how they are doing that. And what you can see here in this movie, you can see that over time, there is one more segment attached. There is one more segment. The way this is growing is because these segments are going. That's associated with rhythmic gene expression that we hadn't seen in humans before, and I think it should work here. There you go. And you can see as the thing elongates, you get this gene expression being patterned, and we can reproduce this in the dish. So I think that this is going to be very useful to study, and we are collaborating with a group with a colleague to try to use this to study the origin of many spinal cord malformations and many spinal cord things. And there is other people that are using this system to generate hearts. Hearts are not just a group of cells that come and give rise to the heart. It requires the integration of many different cell types and cell signals that are very difficult to generate simply by trying to steer everything to make a heart. The heart is the cooperation of many different cell types, and these are called organoids, but they come from embryonic cells and from creating this little blueprint of the organism that allows the cells to interact and to generate the heart. 10 years ago, I did give I looked at this, and I was very funny. I think that's maybe Jochen will remember. That was the title of the lecture, Genetic Alchemy and the Making of Animal Embryos. And the last slide of that lecture was this, that cells are the link between the molecules and the organisms. They are the ultimate alchemists. I think 10 years later, we have a lot more evidence that this is absolutely the case, and that indeed, the elements, the cells are the ones that integrate all these elements and make an organism. And I'd like to finish, coming to the end, with this, I like always, fantastic quotation from Stephen Jay Gould, which to me, it says, I think it's there, and it's, aus reiner Aufregung übertrifft die Evolution als empirische Realität jeden Mythos menschliche Ursprünge Lichtjahre. Ein genealogischer Zusammenhang, fast 1 Milliarde Jahre zurückreicht und nun von Bakterien über den höchsten Mammutbaum bis hin zu menschlichen Fußabdrücken im Mond reicht. Kann das eine Geschichte von Zeus oder Wotan übertreffen? Wenn sich Wahrheitswert und visceralen Nervenkitzel so verbinden, dann liegt, wie Darwin am Ende seines großartigen Buches feststellen, in der Tat Erhabenheit in dieser Schicht des Lebens. And with that, and reminding that it's not just alchemy, I just want to thank you for listening to me, and I look forward to talking with you later.